Hello everyone, I am Dr. Nilanjana Bhattacharya from SBIMS. Today I would like to talk about epistatic interaction. As we all know, there are two types of epistasis, dominant epistasis and recessive epistasis. In this video, I shall discuss the dominant epistasis. So let's start. Epistasis defined as the interaction between different genes. The term epistasis was first used in 1909 by Bateson to describe a masking effect whereby a variant or allele at one locus prevents the variant at another locus from manifesting its effect. So, what do you mean by that? It is simply the phenomenon where the effects of one gene are modified by one or several other genes, which are sometimes called modifier gene too. The gene whose phenotype is expressed is said to be epistatic, while the phenotype which is altered or suppressed is known as hypostatic. So, epistasis can be contrasted with dominance, which is an interaction between alleles at the same gene locus. There are 6 types of epistasis gene interaction, and these are as follows: recessive epistasis, dominant epistasis, dominant inhibitory epistasis, duplicate recessive epistasis, duplicate dominant epistasis, and last but not the least, polymeric gene interaction. So as this video is solely related with the dominant epistatic part, I will discuss different types of dominant epistasis. When a dominant allele at one locus can mask the expression of both alleles, either dominant or recessive, those they are present at another locus, it is known as dominant epistasis. In dominant epistasis, F2 cross yields a typical 12:3:1 ratio. So in other words, we can say the expression of one dominant or recessive allele is masked by another dominant gene. And this type of epistatic interaction is also referred as simple epistasis. So there are two examples. One is coat color in dogs, another is fruit color in summer squash. So if we are dealing with the first example, that is coat color in dogs, among dogs the colors of coats depend solely upon the action of two genes. One gene locus has a dominant epistatic inhibitory allele, that is capital I, of coat color pigment. The allele capital I prevents the expression of color at the hypostatic gene locus, which is either capital B or small b, and they produce white coat color. The alleles of hypostatic gene locus, so here hypostatic gene means any combination of B, either capital B or capital B or small b, express only when two recessive small I occur on the epistatic locus. That is, small I along with two capital B or small I with capital B small b produces black color, and small I with small b produces only brown coat color. And when two such white coat colored dogs are crossed in F1, the white, black and brown coat colors appear in 12:3:1 ratio as shown in the figure. Next, dominant inhibitory epistasis. In this type of epistasis, a dominant allele at one locus can mask the expression of both dominant and recessive alleles at the second locus. And in this case, the F2 ratio will become 13 is to 3 instead of normal Mendelian F2 9 is to 3 is to 3 is to 1. An example, feather color of chicken and anthocyanin pigmentation in rice. So, in case of feather color of chicken, this example used to illustrate the overall coloration of the chicken. The color of a chicken feather is determined by the interaction of two gene loci, capital I small I and capital C small c. Capital I small I here is the epistatic gene. Any chicken with one dominant capital I in their genome will have white feathers. At the hypostatic locus, either capital C or small c, the dominant allele capital C codes for colored feathers, while the recessive small c codes for no color. Hence, a chicken that is homozygous recessive, that is having small c, will also be white. So you can see that our usual Mendelian dihybrid ratio, that is 9 is to 3 is to 3 is to 1, has been changed by the epistasis to 13 is to 3, where among the 16 type, 13 numbers are white color and rest 3 are showing the colored. Next, duplicate dominant epistasis. When a dominant allele at either of two loci can mask the expression of recessive alleles at the two loci. This is also called duplicate gene action. If a dominant allele of both gene loci produces the same phenotype without cumulative effect, that is independently, the ratio will be 15 is to 1. Example, on character in rice. So development of on in rice is controlled by two dominant duplicate genes, either capital A or capital B. Presence of any of these two alleles can produce on. The onless condition develops only when both these genes are in homozygous recessive state. That means instead of capital A, capital B, they are small a and small b. A cross between aunt and onless strains produced aunt plants in F1. Intermating of F1 plants produced aunt and onless plants in 15 is to 1 ratio in F2 generation. The allele capital A is epistatic to capital B or small b alleles, and all plants having one capital A allele will develop on. Another dominant allele capital B is epistatic to alleles capital A or small a. So individuals with this allele also will develop on character. So as I have told you, the on character is totally controlled by the two dominant duplicate gene, capital A and B. In this way, only two classes of plants are developed, and the normal dihybrid segregation ratio 9 is to 3 is to 3 is to 1 is modified to 15 is to 1 ratio in F2 generation. Scientists now know that Mendel's predictions about inheritance depended on the genes. However, unlike the phenotypes that Mendel considered, the majority of phenotypes are affected by more than one genes. Indeed, most of the characters of organisms are much more complex than the characteristics that Mendel studied. And epistasis is one source of this complexity. Epistasis can occur in a variety of different ways and result in a variety of different phenotypic ratio. These dihybrid crosses produce a phenotypic ratio that differs from 9 is to 3 is to 3 is to 1, that is the classical Mendelian dihybrid ratio. And the deviations are, as I have discussed, 12 is to 3 is to 1, 13 is to 3, or 15 is to 1. But one thing you note that each of these modified ratios can be obtained by summing one or more of the 9 is to 3 is to 3 is to 1 classes expected from original dihybrid cross. Thank you.